Hello students, today we are going to discuss in detail on databases of bioinformatics. First of all, we'd like to know what is the importance and scope of the bioinformatics. Bioinformatics is the subject of biology, computer science and information technology. So in this information, what information we are storing means we are storing the information of biological data. What are all the biological data which is stored means the information of nucleic acid, RNA, protein. So in the case of nucleic acid and RNA, we store the information of the sequences of nucleic acid where we store irrespective of the plant species, that is plant. We have separate databases are available for plant, animals, insect, viruses, bacteria, and as well as in the case of the human being, we have all informations are stored in the database. And in the case of the protein, we come across primary, secondary and tertiary quaternary structure of the protein. Databases are also available in the system. This computational technique for biological information was developed by Margaret Dayoff. So here is the scope of bioinformatics, better documentation, and it requires large storage quantity of data. Whether we have to add, if you are inventing a new organism, or if there is any alteration in the sequence or variation in the case of the nucleotide sequence of organisms, that can be stored or documented. If any data is not necessary, that can be deleted, that is possible in the case of this. Why we are storing all this information means in order to discover and design drugs. Considering the genomic structure of the pathogen and chemical structure of the drug, we can develop newer and newer drug, because nowadays the bacteria are gaining multi-drug resistance. They cannot work against some of the antibiotics. So we are in a position to develop newer and newer antibiotics. So such cases, in order to develop antibiotics or new drugs, we are in need of the basic information of the bacteria as well as in the case of the drug also. We can study the important biomolecules such as protein and nucleic acid and mRNA. Protein is the structural and functional unit of a particular gene. So nucleic acid, why we are studying means it is the genetic material that is passed from one generation to the next generation, and it is responsible for various genetic disorders, any diseases, any changes in the nucleic acid sequence resulting in the development of genetic disorder. So bioinformatics is used to compare already available details of protein and nucleic acid, and very easy to search and access information, and fast and accurate for logical analysis and interpretation and prediction. All these are the scope of our database which is available in the bioinformatics. What are the application? We can compare the nucleic acid and protein sequences of different species, and it provides similarity and differences between the sequences of proteins and nucleic acid. There are two types of analysis there, structural analysis and functional analysis. That is why we call it as genomics, transcriptomics, proteomics, metabolomics. Genomics talks about the information of genes that is present in the chromosome, and transcriptomics is talking about the mRNA, information of mRNA, tRNA and RNA, especially transcription, that is conversion of the MR, synthesis of how the mRNA is synthesized from the genetic information which is available in the gene. Then later, proteomics is study about how the message which is present in mRNA is translated to protein with the help of our tRNA as well as rRNA ribosomes. And further we talk about how the protein, which is the product of gene, how it functions, what is the role of it, and where it works. That there comes the role of our metabolomics, in which metabolism the particular protein is associated, or it is structurally associated with the part of the cell or it is some doing some function. So what type of function it carries out, all these details we want to study. So this is the branch what we see. Basically we call it as it is structural analysis and functional analysis. A molecular level of classification of the organisms can be made possible by using our bioinformatics tool. The classification, the comparison of similarities and differences between the nucleic acid sequence and to find the relationship between nucleic acid and the protein. In the taxonomy, morphological enzymatic comparisons are occurs only accurate level analysis are needed. In order to compare, why we are comparing, in order to compare the protein and nucleic acid, we require the classification of protein, classification of nucleic acid, classification of individual, and evolutionary relationship between the organisms. Gene finding. Using bioinformatics, finding the gene is very easy. Nucleic acid is the expression product of the gene. By finding the nucleic acid sequence, it is easy to help and identify the genes responsible for certain characters. Thereby, we can improve it. By finding the gene in application of crop improvement, such as we can able to select or develop the plants which are resistant to insect, disease, drought, salinity, and we can produce higher yield crops. In agriculture and medical field, it is useful in comparison of normal one with the diseased one. In the medical field, we can able to find the gene responsible for genetic disorder and we can rectify in embryo in the embryonic stage itself with the help of embryonic stem cells, and we can make use of the embryonic stem cells uh, for the patient level in the adult level to treat the particular disorder to make the diseased one to become normal one. By embryo therapy at the embryo level itself, we can rectify them when we rectify the mistakes in sperm, egg automatically the baby will be normal, which is disease free, that is genetic disorder free in nature. If the patient is treated particularly at a particular cell level or nucleic acid level, the patient will be disease free by this mechanism. And protein structure prediction, next application where we can compare the structure of the protein and we can find the what is the activity and role of the particular protein in the physiological and metabolic pathway of the organism, we can able to find out. And we can find the disease pathway, how the protein become defective in the generation of the disease. And by identifying the protein coding gene, we can able to cure the genetic disorder. Normally, in order to predict the structure of the protein, we require the techniques like NMR technique and X-ray diffraction analysis are utilized to identify the protein structure, but it is a very expensive and time-consuming method. Whereas in the case of bioinformatics, it is very easy and less expensive and time-saving also. Very short time is required for the structure prediction in the case of the bioinformatics level, and discovery of new proteins can be done using this bioinformatics instead of the normal X-ray and NMR X-ray diffraction and NMR studies, which is helpful in finding the drug and pharmaceutical for it. By knowing the protein structure, we can synthesize biologically valuable enzymes also for the sake of us, for the treatment procedure. And further, it is helpful in identifying the evolutionary relationship among the organisms. By structural genomics and functional genomic comparison are carried out to find the relationship among the various organisms. And for the construction of the biological database. Biological database means where we can store our information of different types of databases of nucleic acid, RNA, protein, etc. and drugs or the therapeutic drugs. The database is the space where the information are stored in the electronic format. So where we store How we store means, if it is in the form of file format, it occupies large amount of space, and moreover it is time consuming also. And protecting these databases in the form of file format is difficult, but it is easy for us to store the information in the electronic format in the database. So example, the information of protein and nucleic acid is available in the EMBL, European Molecular Biological Laboratories, and Gene Bank. The total genomic structural study of an organism is helpful in identification of the species of the organism. And further it is used by finding the gene, we can involve for the bioremediation also by cleaning up the environment. In oil spills, we make use of Pseudomonas butida, which decreases the effect of hydrocarbon in oils, and plasmids that degrades hydrocarbon, total oil degrades, which improves and modify the individual use for bioremediation. And creation of bioweapons. The bioweapons are nothing but our drugs which targets the different disease causing microorganism and to kill them. Now we are going to see the details of the biological databases. So various nucleic acid databases are available, that includes EMBL, Gene Bank. Structure of Gene Bank entries, specialized genomic resources, UniGene. We'll see one by one. EMBL. EMBL is a nucleotide sequence database. It was developed by European Bioinformatics Institute in UK. So EMBL, abbreviation for this is European Molecular Biology Laboratory. It collects the information of different sources such as genome sequencing projects, scientific literature, direct author submission. So if you are doing any project work by comparing two different sequences, you can submit your data directly to your EMBL after authorization from them. It is associated with Gene Bank, DNA Data Bank of Japan for exchanging information for each other, so that we can see the comprehensive collection of information. So the growth rate of this website is very fast, and double information is made possible in 9 to 10 months. It has many subdivisions are available. The laboratory operates in the 6 different cities, such as the main lab is in the Heidelberg, outstations are in Exton, European Bioinformatics Institute in England, and Grenoble in France, Hamburg in Germany, Rome, Italy, and Barcelona, Spain. The EMBL group of laboratory performs basic research in the molecular biology and molecular medicine, as well as training for scientists and students and visitors. They are even providing training how to make use of, how to use their website containing, how to handle the database which is available in their databases. Data which is available in the database. They are providing certification also. Informations are accessed through SRS system, that is how to get information. If at all you want something from a person, means what you are doing, you are going and asking. No, same manner we can ask the information through a sequence retrieval system, SRS system, which is available. Further systematic genetic analysis of embryonic development, the Fruit 5 was conducted by EMBL by Christian and Eric, for which they were awarded Nobel Prize of Physiology or Medicine in the year 1995. In early 1980s, the Jacks and his team, they developed this electronic format of your this one EMBL, and further cytogenic electron microscopy for biological structure were created by them. They were awarded with the Nobel Prize during the year 2017. Here is the URL or the website link, which is given for your HTTP is given for your reference, www.ebluk.embl. This is the URL address where you can go and search the information of this database. Here we can see this is the screenshot of the particular website. Next one is gene bank. The primary nucleic acid sequence of stored in a biological database. The full form is gene bank. It is developed by NCBI, that is National Center for Biotechnology Information. It has less restriction and it is helpful in scientific and research community and supports the research activity where we can uh, where it contains information without a restriction except copyright sequences and patented sequences are not cannot be misused by anyone. The growth rate, it is 1 month. Within 1 month it doubles the information and the informations are divided into 17 divisions for getting the informations easily so that we can it is convenient and efficient to get the information in the gene bank. There are two retrieval systems are available. Retrieval means how to get information or access information. Entrance integrated retrieval system. Another one is Medline facility. Entrance ENTRENZ. Entrance integrated retrieval system. It has the capacity to link the nucleotides sequence database with that of the protein sequence database so that we can compare which gene is converted to a protein. And Medline facility is useful to get information of abstract of originally published papers related to nucleotide sequence in this case. So the website is URL is www.ncbinlmnih.gov/genebank. This is the website link for it. Gene banks incorporates information from published available sources and primarily from direct author submission, and large scale sequence projects are available so that it ensures comprehensive coverage so that it gets the original information from the direct author of the particular project as well as the paper for this published products are available. Even it can exchange the data with both EMBL data library and DDPG. The structural entities which are present. The structure of the gene bank entries includes the gene bank release includes sequence files, indices created on various databases and information derived from the databases. These are available in the form of CD-ROM also. It is convenient mechanism and it is widespread in nature and relatively inexpensive. As the size of the database is larger, the number of CDs required are difficult to handle for the producer as well as the user. So today the gene bank, that was the earlier days format. Nowadays the gene bank is available in the file transfer protocol of the internet services. The commonly used sequence entry file is containing the sequence of itself and descriptive information related to it. The entry consisting of number of keywords that are related to associated with the keywords and an optional features. The structure of the gene bank entry is consisting of 13 structural components such as locus, definition, accession, number, version, keywords, source, organism, reference, author, title, journal, PubMed number, remark and comment. Locus. Locus is we need to provide an entry number for the identification of the nucleotide sequence. Definition is the scientific name of the source organism. For example, Bt gene means it refers to Bacillus thuringiensis, mRNA, B endotoxin. These are all the related definitions that are associated from this. Accession number, that is a normally similar to entry number. Say entry number, and version is the updated information what we have done. First written by the entry number and version also updated. If you are making any changes, that also gets updated in this case. And keywords for the search of the particular data. So just like a keywords when it is given, where we can go and search. And source, the source of the organism where we got, we should write the common name as well as the name of the organism with scientific name also. Reference. The reference of the paper published related to the nucleotide sequence of interest, who is the author, in the order of the author, where it is published. All these things, the title of the paper, journal, in which journal the paper is published by the person, and PubMed number, in which helps to access the archived published paper, in which PubMed journal archiver will be available, and remark or any comment that is given also in association with it. Specialized genomic sources are also available in the case of the system. That includes unique gene. It's a collection of gene from many organisms, and each cluster relating to unique gene and including related information corresponding to the gene is loaded here. The valuable role of Unigene is the gene discovery. It is used for gene mapping projects and scale gene expression analysis. It can be used as a project also by you when you are going for the final year projects. TDP, that is TIGR, is these databases containing DNA and protein sequences, gene expression, protein family information, etc. Also, the data such as a taxonomic range of plants and humans. Role of cellular components are also present. And SGD, Saccharomyces Genome Database, is an online data resource which contains information on the molecular biology and genetics of Saccharomyces cerevisiae or pudding yeast. This data provides internet access to the genome, its gene and its products. SGD database helps the research field by uniting together the function to perform the sequence similarity search tool, where we can identify the similarity in the sequence of one species with that of the other species. Further, we can create genetic map using dynamically created graphical display and make the database as a user friendly. The next database, which is our genomic database available, is Unigene. These are all the database which is tend to be linked and extended, where primary database of DNA is available. The purpose of genomic resource to species specific genomics and particular sequencing technique. The primary goal of the Human Genome Project is to determine the complete sequence of the human genome, that is 3.2 into 10 to the power 9 billion base pairs, and 3% of the genome encodes protein. Already we discussed in our previous video. Biological, some of the protein and gene function where we don't know the biological function need to be explored. A transcript map is a vital source that is flagging the parts of the genome that are actually converted to gene which are expressed to protein, or we call it as gene expression. So the genes which are converted to mRNA by transcription, then later to protein, we call it as gene expression. Okay. Unigene attempts to provide transcript map by utilizing the which genes are undergoing transcription and later to translation to protein, transcription to mRNA and later to protein. So this map is providing, transcript map is providing a set of non-redundant genes, oriented clusters derived from the gene bank sequence. Further, the collection represents genes from many organisms, each cluster relating to a unique gene and including related information such as the type in which the gene is expressed and map location, etc. So far we discussed gene databases, that is EMBL, DDBJ, gene bank, etc. Now we are going to see the protein sequencing databases, that includes PIR, Protein Information Resource, SwissProt, TREMBL, Tremble, and composite protein databases includes NRDB, OWL. Another one is secondary databases. These previous ones are primary databases, and this is secondary database that includes ProCite, Prints, Blocks, and Identify. SwissProt is a protein sequence database. It is developed by the Switzerland-based database. It is developed by the Department of Medical Biochemistry and University of Geneva and EMBL library during the year 1986 to 1987. It is a curated protein sequence are available. It provides high level of annotation. So we can see how the function of the protein, what are all the post-translation modification, what are the variants and sources of the organism. It also provides the details of high level of integration with the other databases. The Swiss plot provides information, name and origin of the protein, what is the genetic information, what is the ontology, sequence, annotations, what is the amino acid sequence, what are all the bibliographic reference, cross-reference with the structure and interaction databases and the entry information. Here we can see the screenshot of the particular web page, Swissplot and Tremble. It is maintained by the collaboratively by the Swiss Institute of Bioinformatics and European Bioinformatics Institute. It contains non-redundant entries and informations are thoroughly reviewed and annotated. It is linked with the other databases like here EMBL and Tremble. Tremble is the primary protein sequence database. It is translated EMBL. Translated EMBL means how the nucleic acid sequence are converted to protein. That is, in short, we call it as translated. First two letters of TR translated. TR is taken, that is added with your EMBL, European Molecular Biology Laboratories. And it is created in 1996 as it is supplements to Swissplot. It contains completed annotated protein sequences. Tremble sequence database contains translation of the all coding sequences. CDS is present in the DDBJ and EMBL gene bank nucleotide databases, and it also sequences are extracted from the literature submitted to Swissplot and gets integrated. This is a screenshot how to get all the information from this Tremble. The Tremble consisting of two divisions, that is SP Tremble and REM Tremble. SP Tremble is a temporary storage area where incomplete sequences which are not yet manually annotated, when it is fully described and contains entries that are eventually been incorporated into Swissplot. Tremble is developed by European Biological Information System. Whereas REM Tremble, it contains completely explained and fully annotated sequences. What is there, what is details, why, what is the function, all the complete details will be present in the REM Tremble. It contains sequences that are not destined to be included in the Swissplot. For example, we can see immunoglobulins, T cell receptor, fragments of 4 then 8 to 8 amino acids, synthetic sequences, patented sequences. All these are available in the REM Tremble. Next database is nothing but PIR, Primary Protein Sequence Database. That is PIR. First alphabets are taken. Primary Information Protein, Primary Information Resource. During the year 1960, it was developed by Margaret Dayoff. It's a collection of sequence for investigating evolutionary relationship among the various proteins. So PIR is the base for the comparison of evolutionary origin containing database of the protein. So developed at the National Biomedical Research Foundation. This is developed, it is further split up into 4 databases, PIR 1, 2, 3 and 4. So based upon the quality of data, they differ in terms of quality of data, level of annotations. So here we can see the classification of PIR 1, 2, 3 and 4. What is their function? PIR 1 contains fully classified and annotated protein database, and PIR 2 includes preliminary entries which have been not thoroughly reviewed and may contain redundancy. and PAR3 contains unverified entries which have been not reviewed, and PAR4 contains protein sequences that are not genetically encoded and are not produced on the ribosome. They are synthetic protein sequences. Here is the composite protein databases. These are all the amalgamation or compilation of products of different primary databases, so that the researcher can search it easy and efficient manner. They render sequences searching much more because they are abbreviated and need to be interrogate multiple resources, that is NRDB and OWL. NRDB is non-redundant database. It is built and locally at NCBI. It's a combination of 6 primary databases. It's a secondary database. So it contains information which is derived from the primary, all the secondary databases. That includes SwissProt, PDB, PAR, GenePept update, SP update. It is non-redundant and error-free in nature. It is making more efficient via avoiding to search too much of database for getting related informations. OWL, Ontology Web Language. Web Ontology Language, it's comprising of 4 primary databases that contains information of gene banks, SwissProt, NRL3D and PAR4. This is making and searching more efficient way of avoiding too much of database for getting the information. It is developed by NCBI. If any redundancy in gene bank, it is much incorporated OWL during the process. The SwissProt has the highest priority. OWL is only released on a 6 to 8 week basis. It was developed by UK in association with Dasberg Laboratory during the year 1994. Further, we have secondary databases that includes ProSites, prints, blocks and identify. They contains fruits analysis of the sequences which are found in the primary database. It is a shortcut. Always the secondary databases are shortcut because need not to search in larger level in the huge data which is available in the primary database. ProSite, generated by the primary database of SwissProt. It is produced and maintained by SIB. The URL address for it is the www.prositeexpressy.org. It contains group of proteins which are grouped into different families based upon the single conserved motif. Motif is a ring of amino acids. Nearly 10 to 20 amino acids will be present. They are responsible for the protein function and preserve the 3-dimensional structure of it. Such motifs can be easily identified. They are responsible for the biological functions. They are found in the case of enzyme active site, ligand or metal binding sites which are found on it, that can be identified by this method. So they are called as signature sequence of the protein family. We can identify such motif signature sequences by using this database ProSite. The ProSite is developed largely by the manual process for the seeking pattern that best fits particular family and function. It has entries developed from the SwissProt and it is available. The documentation files provide details of the characterized family, biological molecule of the chosen motif, supporting biography also. The significance to find the families based on the motif, how one family of organism is associated with the other one. Further, it is responsible for characterization and annotation of the protein sequence. It is useful in identifying possible function of newly discovered proteins. It offers tools for protein sequence analysis and motif detection. It is part of Expressy proteomics analysis server. And moreover, This is useful in the classification of protein based on the highest conserved motif. It is also useful based upon particular motif that can identify the characteristic features and represent the characters. We can use this is useful in finding the structural and functional details of the protein which are present in the form of motif structure, which is of shorter in sequence that we have seen already 15 to 20 amino acids. The next one is prints. It's a collection information from the owl. It collects the information from the SP, Tremble and Swissplot. It is contributed by SAB, and here is the web link HTTP, that is URL link is provided for it. It was created during the year 1999 by the Department of Biochemistry and Molecular Biology at University of College London UCL. Here we need to consider multiple motif and insert into the single common motif. We require a more accurate analysis based upon the similar multiple motif shared by sequences. Another one is called as blocks. There are multiple motifs based databases. Uncapped multiple alignments of motifs are available. Databases are constructed by using automated proto-match system. Nearly 99.5% of the matches are true and are true negative in nature. Second median value of these true positive scores are shown. Two scores are noted for each block of the particular motif in this case. It's a fully automated method of database system that is shown here in the case of the ProSite. The information which is derived from secondary database, prints and ProSites can be called as tertiary database. It is based on the protein family ProSite. The motifs or the blocks are created automatically, and we can identify how they are useful in detecting highly conserved regions of each protein family from one species to the other species. The blocks are ultimately coded. They are identified by, each block is identified by ID line and accession number. ID line indicates the, while we are searching, the entries are, ID line indicates the type of discriminated expect in the life. AC line indicates the minimum and maximum distance of the blocks from its neighbor, and DI line contains the description for a title of that family, and BL indicates the diagnostic power, amino acid triplet, number of sequence. All those details are present in the basic line, or what we call it as BL line. Identify is another automatically derived tertiary source from blocks and prints, which was developed by the biochemistry at Stanford University during the year 1998. They are called as E-motif. So it is based on similarity of highly conserved motif. That is, some of the proteins are conserved in nature during the course of evolution. For example, cytochrome P450 from one organism to the next organism, from the lower prokaryotes to the higher eukaryotes. So such E-motifs are databases are stored in the case of this identify. This database is constructed on generalized expression of similarities in the highly conserved motif sequences. Further, we have the classification of the database that includes structural similarity and reflecting the evolutionary origin. Structural similarity means where we can predict the primary, secondary and tertiary structure of the protein. From the primary we can develop secondary and tertiary structure by using this SCOP and CATH. SCOP is structural classification of protein. It is maintained by the MRC Laboratory of Molecular Biology It is used to describe the structural and evolutionary relationship of proteins between various organisms. It was developed in the year 1995. It is constructed using combination of manual inspection and automation methods. Both manual data will be there and automated data is also available. And the scope is having hierarchical fashion of the reflects the structural and evolutionary relationship among the various organisms that includes family, superfamily, and fold. Families proteins are clustered into family with clear evolutionary relationship. If they have sequence identity more than 30%, please note down more than 30%, if the sequence amino acid sequence are similar, they are coming under family. Superfamily, the proteins are placed in superfamily in spite of low sequence identity structure. Structure and functional characteristics are common of evolutionary origin, and further we classify them under fold with major secondary structure with the same arrangement with necessary topology structures are available in the case of this. Here is the web link available for that, URL web link available for it. CAT, Class Architecture Topology and Homology. This is hierarchy classification of protein structure maintained at the University of College of London during the year 1997. There are 4 different classes are available in the case of CAT. Subclass 1, 2, 3 and 4. Subclass 1 maintains similarity in alpha helix, 2 similarities in beta sheet, 3 in the alpha and beta sheets of the protein, and 4 is based upon the secondary structure content of elementary and secondary structural elements. That is very less in amount. Architecture, describe the gross CAT. We have architecture, which is the gross arrangement of the secondary structure, and topology is overall shape and connectivity of the secondary structures are shown in the protein, and homology is the 35% sequence identity, 30% or 35%. Here 35% sequence identity means amino acid sequence which are similar, amino acids are similar and they should share a common homologous or sister chromatid similarity in the sequence comparison process by the comparison algorithm. And last one is sequence, the final level in hierarchy. The structure with homology groups are further clustered on the basis of the sequence identity, where we have the sequence identification which are more than 35% similarity in the structure and function by this CAT. Database searching. How to search these databases? Here the sequence database searching involves EST search and different approaches to EST analysis: mark image, insight, TIGR, TIGR, EGAD, EST analytical tools, sequence similarity, sequence assembly and sequence clustering. EST search, expressed sequence database search, where we can able to identify the short nucleotide sequence of cDNA, that is complementary DNA, which is synthesized from mRNA by the reverse transcriptase enzyme. This contains 200 to 500 bases in length that are useful, and some of them theoretical length of single run of 1000 base pairs also. It is called as gene transcript, where we have the information of the gene or mRNA. The EST alphabet is in 5 characters ACGTN. Normally this is used in identification of the particular gene, mapping of the gene by the genome, identification of the species, and for the academic analysis and commercial exploitation have been developed. There are various approaches by which we can do the EST analysis. That includes EMBL data library, Merck Image, Insight, Tigger, and EGAD. Merck Image, this is developed by the company Merck company in the University of Washington. The aim is to produce 3 lakhs EST from cDNA library. They are useful in the production of the drug index. They are helpful in supporting the academic analysis. The commercialization of the expressed sequence tag information for the drug production that is done by them. Their drug index known as Merck Gene Index is developed by them. Insight is a pharmaceutical company. They produce database of life sequence. They contain information of the cDNA library. They are ready to provide information. You can provide and collect information of a relative copy of number of genes in the healthy and diseased tissue. So it is a commercialization of genetic information regarding the EST of healthy and diseased cell, where we can provide the therapeutic targets. The production of the drug for this to get money. In the April 1998 only, the size of the life sequence was about 2.5 million EST. Now it represents 8,000 to 12,000 different genes. Another one is Tigger. Tigger is Institute of Genomic Research that provides the government organization. It is providing information of the structure, functional, and comparative analysis of genome and gene products for the academic purpose as well as for the research. The range of organisms are covered by Tigger include viruses, bacteria, eubacteria, pathogenic bacteria, archaeobacteria, eubacteria of plant and animals. Aim is to prepare human gene index in association with our Tigger, in association with your EST for the over 3000 cDNA library. It is created to view all the human gene of information that their expression pattern, cellular load and function and evolutionary relationship. EGAD is expression gene anatomy database. It provides information of the EST. There are many tools available for the EST. The commercial available tool is Insight Life tool. Publicly available tool is 3 types: sequence similarity search, sequence assembly, and sequence clustering. These are all the sequence similarity: BLAST tool, BLAST P, BLAST N, BLAST X, and X BLAST N. Sequence assembly, where we can assemble the data, where we can align and merge the different fragments of sequences to reconstruct the original mRNA. Sequence clustering, these are all the programs takes larger set of sequences and that divides into subset and clusters. Sequence similarity searching tools, they make use of, there are 2 types: pairwise sequence alignment, similarity sequence searching tool. Example, BLAST and FASTA. Homology sequence research is done by Clustal and Modeler. SciBLASTA, BLAST, FASTA, SciBLAST. Local sequence alignment is done by this global sequence alignment, another tool. That includes GOFA, Ermine, and Introstan. Structural analysis of nucleotide and proteins are done by SwissProt, PDBviewer, and RASMOL. Statistical analysis tool to find the similarity and differences. Value can be done by Statistica, MATLAB, and Perl. And pairwise sequence alignment is done by PALM, substitution matrix, that is identity and similarity matrix, that is known as similarity matrix, and substitution matrix of PAM, percentage accepted mutation, percentage amino acid mutation, BLOSSOM, dot plot, BLAST, and FASTA. Here is a substitution matrix. BLOSSOM and PAM is used to compare the sequences of amino acid from two different sources, which is of to study the evolutionary relationship among the organism. If any amino acid is changed in nature, that is taken into consideration, and whether the changed amino acid is of similarity in function, chemical structure, or something else. Blossom is block substitution matrices, where block by block we analyze it. Here the blossom has matrix 62. We have 30% similarity, 30 to 62% similarity, and 62 to 90% similarity that are given other versions of this comparison. Blossom 30, 62, 90, that indicates how much of percentage of similarity is available. And PAM is point accepted mutation that was created by Dayoff. She was a lady. She derived amino acid substitution matrix where she compared the sequence of homologous sequences of protein during the evolution, how these substitutions do not find significant changes in the function of the protein, how they are accepted. They are accepted naturally and selected during the course of evolution, according to Darwin's theory. These matrices are known as accepted point mutation or point accepted mutations. So in order to prepare the PAM matrix, we require 20 into 20 mutation probability matrix. So there are different types of point mutations are available. No mutation, because in the codon table any amino acid, that is the nucleotide change, may result in the same amino acid. For example, when G is changed to A, it represents the same lysine because the codon is degenerate in nature, and silent, that is the same amino acid. Even though the mutation has happened, but the same amino acid is occurring due to degeneracy of genetic code, we call it as silent mutation. Another one is nonsense mutation. When A is changed to U, that time the UAG is a stop codon. And missense mutation means TTC becomes TCC, the DNA level that reflects in the mRNA AGG complementary, and protein where we have, instead of lysine, we will come across arginine, that is a conservative. Non-conservative missense, some other amino acid comes over here, TGC, ACG, threonine comes over here, totally different amino acid comes and occupies. But lysine and arginine are similar in the function. Both of them are basic amino acids, but threonine is a hydroxy amino acid. We call it as missense mutation or non-conservative missense mutation. So such mutations can be compared, and how some of the amino acids are accepted during the course of evolution, some are not. We can analyze it, and further in the case of the blossom, we go where in a PAM we go individually amino acid by amino acid, whereas in the case of the blossom we go block by block. And the basic method is nothing but a dot plot analysis, where we write the sequence and we find if there is the sequences are similar, we put it as a dot. If not, means we never mark anything. This was developed in the year A.J. Gibbs and G.M. Memory during the year 1970. What the other methods are the advanced version, that is other matrix. Here we can see the dot plot. This is used for improvised logical sequence analysis, comparison of protein sequence analysis, nucleic acid sequence analysis, etc. Last is the basic local alignment searching tool that is associated with our NCBI, where we can see the local sequence alignment instead of the whole sequence alignment by using this BLAST technique. Here we can see the BLAST search of nucleotide as well as protein. their merits and demerits, and this the types of our blast P, blast N, and T blast N, blast X, and T blast X. FASTA is another format. FASTA format is a text-based format representing the nucleotide sequences or amino acid sequences. We have different forms of the FASTA is there. FASTA A3, FASTA S3, F3, XY3, and TFAST XY3. Multiple alignment techniques are also available. Databases of multiple alignments can be done with the help of SciBlast and ClustalW. Many sequences can be analyzed simultaneously instead of two sequences comparison. More than two is made possible by this multiple sequence alignment method. In short, we call it as MSA. They are fast and advantageous and very accurate. In progressive multiple sequence alignment, where we can progressively step by step we can analyze, but it is time consuming when compared to the previous one. Hierarchically we can study it, and evolutionary relationship is made possible in this progressive multiple sequence alignment that is not possible in the conventional alignment. SciBlast is position specific iterative basic local alignment search, PSSM. That is position specific scoring matrix of the protein blast is done by this. The SciBlast is most convenient and used by the internet with the help of the graphical user that is provided by the NCBI. This PSSM matched by this method very easily by using this SciBlast. That's all about various databases which are available in the case of the bioinformatics. Thank you.